Assalamu alaikum. Welcome to Dr. Nerd. In this video, we will be discussing the primary structure of a protein and the secondary structure of a protein. On a separate video, we will be discussing the tertiary and the quaternary structure. And on another separate video, we will be discussing the clinical aspect of improper protein structural formation. So I'm pretty sure you all are familiar with the basic backbone of all amino acids. In the center, see, all amino acids have a central carbon. All amino acids have a hydrogen over here. All amino acids have an amide group. And all amino acids have a carboxylic acid group. Right? Now, these things I've mentioned are common amongst all amino acids. This R group is different from one amino acid to another. Right? This is what makes each amino acid different. Everything else is the same, but the R group is different from one amino acid to the other. So let's get into the bulk of this video. We'll start with the primary structure. Right, so on the left-hand side you have valine. The right-hand side you have alanine. Notice how they both have the central carbon, the hydrogen, the amide group, and the carboxylate. The only difference is the R group. In this case, in alanine it's CH3, and in valine it's this big thing over here. So these two are two separate amino acids, and we want to combine them together. Now, how do we do that? Well, first off, we remove an oxygen from the carboxylate, and so we're left with carbon and oxygen here. And see this H3? From it, we remove two hydrogens, and so we're left here with HN. Now, notice how these two, oxygen and H2, if you combine them together, they form water. Now, this is what is removed in this process of combining two amino acids, right? For this reason, we call it a dehydration reaction. People will also refer to it as condensation reaction sometimes. They're both the same thing. This is just simpler to understand. Now, you're not actually removing water. You're removing oxygen and two hydrogens, which together form water. Now, you remember how here... Right, so remember how here we were left with carbon and oxygen, and on this side we were left with hydrogen and nitrogen. Now these two will form a bond between each other. Notice this bond over here? This is what we call a peptide bond. This whole thing is what we call a peptide bond. Right, this is the same carbon oxygen as this, and this is the same hydrogen nitrogen as this. And then between carbon and nitrogen, you have this bond. Now, peptide bonds are said to have covalent bonds. And if you remember from high school chemistry, covalent bonds are bonds in which two elements share electrons. In this case, carbon and oxygen, and nitrogen, sorry. So this is a double bond basically between carbon and nitrogen. Two valence electrons are being shared. Now, I'm going to draw the same peptide bond over here. This is the peptide bond. I want to compare it to these two over here. This here is a single bond between carbon and nitrogen. Now, single bonds tend to be long and weak. Here you have carbon and nitrogen in a double bond. Now, double bonds are short, but they're strong. So, what type of bond is a peptide bond? Well, it's actually neither. Peptide bonds are neither long single bonds nor are they double bonds. They are what we call partial double bonds. Now, why do we call it this way? Now, you see these valence electrons here between carbon and oxygen? Apparently, for some reason or the other, they interact with the electrons here between carbon and nitrogen. So, and this interaction allows the peptide bond to have partial double bond activity. So it's not a true double bond, but it resembles a double bond. For that reason, we call it a partial double bond. Now, partial double bonds are shorter than single bonds, but they are longer than double bonds. Now, with the same token, peptide bonds are stronger than single bonds but weaker than double bonds, right? It's a partial double bond. So now that we have this big thing over here, we have this carbon oxygen from here and hydrogen nitrogen from here forming a peptide bond. We've removed water. Now we have what we call this polypeptide molecule, right? Now, how do we name this thing? First, you start off with the amino acid on the left. In this case, is valine. And it just happens to be that the amino acid on the left has its amide group unchanged. Notice how this amide group is the same as the amide group over here. And so we begin the naming process with this amino acid on the left, because amide group remains unchanged. And the first amino acid has its last suffix changed to YL, so valine becomes valile. And the last amino acid, the amino acid you end the naming process with, is the amino acid on the far right. In this case, obviously, is alanine. Now, alanine, which is the last amino acid, has its carboxylate unchanged. And this is the case with all amino acids on the far right. Their carboxylates are always unchanged. Now, there's something more about the peptide bond I forgot to mention, is that they're said to exist in what we call a trans configuration. Now, what does that mean? So this amide group here from the alanine, notice how it's facing downwards. Then you have this amide group here from valine. Notice how it's facing upwards. They are on different directions. One is facing down, one is facing up. Because they exist in different directions, these two R groups, did I say my groups previously? I meant R groups, sorry. So the R group from alanine is facing downwards, while the R group from valine is facing upwards. Now, because these two R groups are facing opposite directions, we say that they exist in a trans configuration. Now, if CH3, for instance, existed up here, it would be just adjacent to the R group of valine. In this case, we would call it a cis configuration. But in reality, the R groups are in opposite directions, so we call it a trans configuration. Now, all peptide bonds exist in trans configuration. Now, see this process of dehydration? Now, it doesn't only occur once, it usually occurs multiple of times. So, in continuation to the valile alanine, you'd have a peptide bond here, and then an amino acid here. Peptide bond, amino acid, a peptide bond, and amino acid. This process continues until you have hundreds or even thousands of amino acids linked together via peptide bonds. So you end up with something like this, right? Notice how many amino acids are connected via this red thing here, which is the peptide bond. Notice how the first amino acid, in this case leucine, has its amide group unchanged. The last amino acid, isoleucine, has its carboxylate unchanged. Now this structure here, which is basically many amino acids connected in a line, right, by multiple peptide bonds, this is what we call the primary structure of a protein, right? So again, it's a line, many amino acids, and connected via peptide bonds. This is what we call a primary structure. Now we move on to the secondary structure. This is the same primary structure we ended up in the last slide. Now this is going to undergo some changes. What sort of changes? Well, amino acids in this chain will acquire hydrogen bonds. So for instance, see proline over here and methionine over here. Now these two amino acids are relatively far away from each other. They can't form bonds with each other because they're far away. So what happens is that this whole thing, right, that I've circled, changes its structure to look like something like this. Now what is the benefit of this? See proline over here, methionine over here. Notice how right now they are close to each other. Previously, they were far from each other, so bonds between them was impossible. But in this case, right, you changed this whole structure into what we call a spiral structure. And so proline and methionine are now close to each other. Now they are capable of forming bonds with each other. Now the bonds exist between the hydrogen from the amino group of methionine and the oxygen from the carboxylate group of proline. Notice how they form a bond, hydrogen-oxygen. This is called a hydrogen bond. Right? Now this structure, this spiral structure, is called an alpha helix. Right? Notice how here I've drawn an X, because this bond shouldn't exist. Right? Because first of all, lysine isn't on the side. Only amino acids on the side over here, over here, over here, and over here can form hydrogen bonds. Leucine is not on the side, so it cannot form hydrogen bonds. Another mistake in this bond, right, is that notice how the hydrogen bond is not parallel to the structure of the alpha helix. And that is a property of hydrogen bonds and alpha helices. They exist in parallel to the structure of the alpha helix. Notice how this is going from up and down, and the alpha helix also up and down. And so they exist in parallel. So this is completely wrong. So you should focus on this one, which is more accurate. As I said, this is called an alpha helix, characterized by changing of the structure into a spiral structure to allow amino acids that are 3 or 4 residues away from each other to form these hydrogen bonds. And this hydrogen bond is really there for stability, right? Now, one hydrogen bond by itself is weak, but you see you'll have many hydrogen bonds between many different amino acids. So collectively, these bunch of hydrogen bonds will work to stabilize the structure of the alpha helix. And this is important in the process of protein structural formation. You need stabilization, you need strength. Hydrogen bond is one way to achieve that strength. It's the only way in forming secondary structures, actually, because alpha helix is a secondary structure. There's another secondary structure we will come to shortly. Notice how in this table now, we have many amino acids, proline, glycine, glutamate, tryptophan, valine. Now they have low alpha helix propensity. That means they rarely participate in forming alpha helices, right? Because they disrupt the structure of the alpha helix. So this really shouldn't be proline. I'm sorry for writing proline here. It should be anything else, really. I don't know, serine maybe. It could be any other amino acid. Could be aspartate, right? Could be any other amino acid except for these amino acids in this table. Because these amino acids do not form alpha helices because they disrupt the structure. They destroy the structure of the alpha helix. So alpha helix is one type of secondary structure. And as we said, it is when this primary structure, it sort of changes shape. When it changes shape, it creates this secondary structure called an alpha helix. Now, beta sheets is another example of secondary structure. Now, there's a huge difference between beta sheets and alpha helix, obviously. Now, one major difference is that, notice in beta sheets, we have one chain of amino acid here and another chain of amino acids here. So, you have 2 separate chains of amino acids, of polypeptides. They're connected, the same hydrogen bond, but this hydrogen bond connects two different chains. And in the alpha helix, the hydrogen bonds connected amino acids of the same chain. Because you see, proline and methionine were in the same chain. After forming the alpha helix, they're still on the same chain, and they form hydrogen bonds, right? In beta sheets, you have 2 different chains, and they're connected via this hydrogen bond. Now, beta sheets exist in 2 conformations: parallel, right, and anti-parallel. Parallel is when the first chain moves from the amide group to the carboxylate, the second one moves from the amide group to the carboxylate as well. So they're parallel to each other. They resemble each other, and that's they go from the same direction, from the amide to the carboxylate. So a parallel beta sheet is a beta sheet whose two chains of amino acids exist in parallel to each other. Anti-parallel beta sheets, which is this, notice how the first chain goes from the amide group to the carboxylate, the second one comes from the carboxylate to the amide group. So they're like opposite in direction. So we call it an anti-parallel beta sheet. Everything else is the same. We have the hydrogen bonds. We have two separate chains, but they are opposite to each other, and so we call this an anti-parallel beta sheet. So this was the second type of secondary structure. Remember, the first type was an alpha helix. And another important thing, beta sheets are said to be pleated. Now, when I personally first heard this word, I did not understand what it meant. Then I looked at the structure over here. See how it's going? There's no way to explain this. I mean, you can see how the direction of the plates of the beta sheets, right? It's going sort of up and then down and then outwards and then inwards, or so this shape is called a pleats, right? There's no way to explain it really. I mean, this is called a pleats. When it goes right upwards, downs, upwards, downs, upwards, downs, or outwards then inwards, and then outwards then inwards, and outwards and inwards. Right, this is called a pleats. So all beta sheets, both parallel and anti-parallel, are pleated. And now beta bends aren't exactly a type of secondary structure. They are used in secondary structures. And what sort of secondary structure are we talking about? The anti-parallel secondary structure, beta sheet. So this is a beta turn. Then the function of the beta turn, obviously it can be called beta turn, you can call it reverse turns, you can call it beta bends. The function of the beta turns is to connect two chains that exist in an anti-parallel beta sheet conformation. So you have this chain over here, you have this chain over here. They're connected, right, via hydrogen bond. And obviously because they're two different chains connected together, we call this a secondary structure. Now what type of secondary structure? A beta sheet. Why beta sheet? Because you have two different chains. And they're anti-parallel because notice how here we have carboxylate, on this side we don't have carboxylate, we have amide. So these are anti-parallel chains that exist in a beta sheet. And this is the function of beta bends. They connect two anti-parallel chains. They will not connect to parallel beta sheets. They will not connect an alpha helix to a beta sheet. They only connect two chains of amino acids that exist in an anti-parallel fashion in beta sheets. Now, beta bends essentially are made of amino acids. So this is amino acid, this is amino acid, this is an amino acid, this is an amino acid. And these amino acids typically will have proline and glycine. And the amino acids in a beta bend are connected via peptide bond, the same peptide bond we've discussed earlier. And the first amino acid and the last amino acids, they have a hydrogen bond connecting them. So in the beta bend, the first amino acid and the last amino acid are connected via hydrogen bond. So the hydrogen, right, in here from the amide group and the oxygen from the carboxylate group are connected via a hydrogen bond. Now again, beta bends are a series of usually 4 amino acids whose job is to connect two chains that exist in an anti-parallel fashion in beta sheets. So that was it. We'll see you in the next video when we discuss